The body's immune system is responsible for defending against foreign organisms or pathogens. It has two main components: innate immunity and adaptive immunity. Innate immunity is the first line of defense and involves non-specific defense mechanisms that are activated immediately after a pathogen is detected. It includes physical barriers such as skin, chemicals in the blood, and white blood cells that attack and destroy the pathogen. Cell types involved in the innate response include phagocytes, such as monocytes, macrophages, neutrophils, natural killer NK cells, and dendritic cells. Adaptive immunity involves more complex defense mechanisms that are activated when the innate immune response is insufficient. The response of the adaptive immune system is delayed, highly specific, and long-lasting. Adaptive immunity can be further divided into two types. In the first type, cell-mediated immune responses are dependent on the T lymphocytes. This occurs inside infected cells. The pathogen's antigens are expressed on the cell surface or on an antigen-presenting cell. Helper T cells release cytokines that help activated T cells bind to the infected cell's MHC antigen complex and differentiate the T cell into a cytotoxic T cell. The infected cell then undergoes lysis, which is the breakdown of a cell caused by damage to its plasma. The other type of adaptive immunity is humoral immune response, mediated by B cells and antibodies. With assistance from helper T cells, B cells differentiate into plasma B cells that can produce antibodies against a specific antigen. B cells respond to antigens from pathogens that are freely circulating or outside the infected cells. Antibodies produced by the B cells bind to antigens, neutralizing them or causing lysis or phagocytosis, the cellular process for ingesting and eliminating cells. Innate and adaptive immunity can potentially interfere with gene therapy. Innate immunity can produce injection site reactions, such as redness and fever, but these are typically mild and transient and unlikely to impact factor expression. Innate immunity may also lead to liver toxicity and subsequent elevations in liver enzymes, which may contribute to reduced expression of factor. The impact of adaptive immunity may be twofold. Previous exposure to AAV may result in a residual supply of antibodies capable of neutralizing the transgene vector. Following vector infusion, activation of both cell-mediated and humoral adaptive immunity may result in delayed but more specific responses. AAV capsid can activate T cells that lead to liver toxicity and subsequent reduction or loss of expression. In addition, B cell activation likely results in the generation of vector antibodies, leading to loss of gene expression over time as expressing cells are identified and eliminated. Both innate and adaptive immune responses are reduced by glucocorticoids. The prophylactic use of glucocorticoids and other immunosuppressive drugs to reduce liver toxicity is being tested in some gene therapy for hemophilia clinical trials.